Hello, everyone, and welcome to today's webinar, Beyond Metabolism, Expanding the Horizons of Pharmacogenomic Testing with NGS. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Jonathan Stein, CSO, Medical Director, Tesis Biosciences. Jonathan, you may now begin your presentation.
Thanks, Michelle, and thanks everybody for listening into this webinar. So my name is Dr. Jonathan Stein, and I am the Chief Science Officer and Medical Director for Tesis Biosciences. And today I'm delighted to talk to you about pharmacogenomic testing, what it is, how it works, and sort of where it's all going, and why it's important. So let me jump right in. First, let me give you my backstory, and this is sort of my little disclaimer slide. So this is my background. I guess it's also my conflict of interest. I like all these guys. These are the places I've worked. I'm basically a small town molecular geneticist. I'm from a place called Los Angeles. Maybe you've heard of it. And I have done a lot of R&D and a lot of molecular genetics in my day. Tesis Biosciences is a cancer and molecular diagnostics laboratory consortium based in Colorado, Texas, and Arizona. And we do a lot of testing. And what I'm going to talk to you about today is, again, the role that pharmacogenetics plays in diagnostic testing and its kind of utility and sort of future applications. I should also thank Thermo Fisher for hosting this webinar presentation, which is absolutely lovely. We use some of their stuff for some of our stuff, and it works pretty great for us. And that's pretty much the only thing you're going to hear from me on that topic today. So let me start by saying that when people originally looked at the interaction of genes and diseases, the historical estimates for them was thought to be kind of 8 to 20% on the high end, more for some diseases, less for others. But recent studies have shown, this is a huge population study using US insurance database data, where they took relatedness information from about 56,000 twins, monozygotic or dizygotic, identical or sibling twins, and 724,000 sibling pairs living together or in separate zip codes to look at environmental effects. They found that the heritability for genetic involvement in diseases runs on average about 40%. Again, it's different for different diseases. But where I was going with this is that genes play a much larger role in disease processes than previously thought, and that means that getting genetic information is that much more important in terms of making clinical decisions, making clinical diagnoses. So this is a know your audience question. I know not everybody here is a molecular biologist, so I just wanted briefly to say that there are many types of genetic variations, but we are mostly discussing single nucleotide polymorphisms, which are sometimes called SNPs. They're single base pair changes. A lot of times they don't have any clinical relevance, but in some cases, in some genes, in some sequences, a change in a single set of base pairs will result in a change in the shape of the protein that the DNA is the blueprint for. And because of that, it may impact protein function. And the protein function we're interested in is the ability of your body, or these particular proteins, that have a role in metabolizing medications. That is to say, they're breaking down drugs, so your body can excrete them, or activating them so your body can use them. And both of those things are very important. In terms of terminology, you'll see a couple different ones used. There's pharmacogenomics, which is the study of how variations in the human genome dictate a person's response to medications. And again, the purpose of this kind of testing is to determine the risk of side effects or the likelihood of effectiveness of a given medication. Pharmacogenetics typically refers to a limited number of genes, often involving drug metabolism, and pharmacogenomics is sort of this bigger study of how complex multi-gene patterns work within the genome. I'll argue later on that these are just going to merge. We're just going to get a lot more genetic information, and so it'll be more of a whole genome study. You'll often hear them used interchangeably, though, really. There's also precision medicine, which is a form of medicine that uses information about a person's own genes or proteins to prevent, diagnose, or treat disease. This is also sometimes called personalized medicine or precision health. It is really well implemented in cancer treatment at this point, where information about they'll sequence a person's tumor, and this will help to make a diagnosis, plan treatment, find how well the treatment is working, or make a prognosis, to see what the predicted outcome is of the disease. I also want to briefly discuss therapeutic failures and adverse drug reactions, because they are going to play a really important role in the relative value of pharmacogenetic testing. So ADRs, adverse drug reactions, right? Negative side effects or adverse effects from treatment. How common are they? Well, in one large study, back in the day, they looked at 3,700 hospitalized patients in Britain, and about 14.7% experienced one or more adverse drug reactions, with each additional medication increasing the likelihood of an episode by about 14%. ADR has directly increased the length of stay in about a quarter of patients that experienced it, and the drugs most frequently associated with them include diuretics, opioids, and anticoagulants. In the US, about 2 million people suffer them annually. It accounts for about 7% of all hospital admissions. Serious drug toxicities cause over 4.5 million emergency room visits per year and 100,000 deaths annually, with costs estimated to be $30 to $100 billion a year. Risks of ADR susceptibility, it's not just genetic, they include someone's immune system, again, their genetics, their demographics, age and gender, physiological and exogenous factors such as drug-drug interactions, and diseases and disorders such as renal failure or liver failure, which might impact a person's ability to process drugs. Contrary-wise, a large numbers of studies have looked at the healthcare savings from implementing PGX, and they range from about, so if you do pharmacogenomic testing on people before you give them medication, the savings range from $3,000 to $10,000 per patient per year. And I'll go into a little bit more detail about that part later, but I guess where I'm going with this is this is common. This problem is a very common problem. Therapeutic failure is when a medication doesn't work. And it is a huge problem in psychiatric pharmacology, with about 40% of patients non-responsive to any particular antidepressant medication, 20% of patients non-responsive to any particular antipsychotic drug. And though there is relatively little information about this issue in terms of birth control, we know, I have 6 aunts, and boy, did they give me an earful when I asked them about this. But the average woman tries 3 methods of contraception by age 40, and by 30% try 5 or more methods by that age because of side effects, because they do not like how they feel on these medications. Contraceptive failure rates run 1 to 10% when they look at normal versus perfect use. So I just put 5% up here. So this is also another area where there is a lot of opportunity for improvement. The sort of canonical example of therapeutic failure, by the by, is, oh gosh, women who get pregnant on birth control. So here's a very brief overview of PGX. Basically, it's specific to the medication, it's specific to the patient, right? It's individualized medicine. And basically, for any given medication, someone gets binned as either fast, slow, or sort of an average metabolizer. You'll see the nomenclature bounces around where sometimes intermediate gets used as someone who is a slightly slower metabolizer. You also hear ultra fast and ultra slow metabolizers. And again, these vary by drug and interaction pathway, and there's websites like ClinGen and Univar where the sort of clinical applications and clinical utility for any given gene can be identified. I come at this as a geneticist, so that's sort of where we, go to look at sort of research aggregations to see what's going on. So there's a couple different ways to do this technology. We use NGS. NGS has a ton of applications. You can use it for whole genome sequencing or whole exome sequencing or targeted panel sequencing, as you can see up here. We kind of focus, because only a limited number of genes are currently identified as being clinically relevant in terms of PGX testing, we'll typically do targeted panel sequencing. So there are many applications for NGS: hereditary disease detection, risk modification for common diseases, detection and treatment strategies for circulating tumor DNA. People can also do very early detection of birth defects through circulating fetal DNA, detection of common pathogens in fluids. We just sequence whatever's in the fluid and see what kind of bugs are in there, and also pharmacogenetic screening for dosage and treatment optimization. Yeah. So the application we use here um, is to screen these kind of discrete panels for genes that are known to interact with medication. Um, Again, the risk varies across a number of different demographic factors, but the reality is we're going to check in each individual patient what's going on with them. For some people, for example, you can see the you have this like large population of people kind of in the center of this picture, and they all get the same diagnosis, and the current methodologies, they get the same prescription. But for some of those people, see the sort of green one in the upper left corner of that picture, the drug is going to be toxic, but they may still get an effect from it. And in some cases, you have a yellow group, the drug isn't toxic, but it's not going to help them at all. And in some cases, the drug will be toxic but not beneficial, or not toxic and not beneficial, like every possible combination. And the goal of this PGX testing is for any given medication to identify where you or where the patient is so that they can get the correct medication. The key point on this slide is that bottom sentence there. 15 to 25% of patients have a clinically relevant variant for any given drug. This is common, super common. And if you've got somebody on 5 drugs, the odds are really good that one of them is either contraindicated or at an incorrect dosage based on their particular genetics. So again, you've got these various bins that people go into, and the nomenclature varies a little bit. So here they use extensive or what are called normal drug metabolizers, and so there are these things called wild type or normal alleles. People try not to use normal to stigmatize people who have variant alleles, but again, your wild type is sort of reversed to like an average genotype. And so these folks are going to have a normal metabolism and won't need any dose modification, and this drug is fine for them. And then, and this is 75 to 85% of people. Again, it varies for each drug. It varies by country of origin, patient population, gender, those sorts of issues. Then you'll have other people who fall into a group of intermediate metabolizers, and that might be 10 to 15% of people. And so they might have one variant copy. They might have one wild type or normal wheel, one variant copy of the wheel. And so they may require a lower than average dose for optimal therapeutic response. And then you might have people who are poor metabolizers, where for any given gene, they only have the variant alleles or a mutated allele or a deletion in that allele. So in those cases, they will tend to accumulate the drug because they don't process it very quickly. And so for them, they might internally be experiencing a much higher dose than, say, a normal metabolizer. They break down the drug much more slowly, and so it's accumulating in them, right? And so they might be getting 20% more or 100% more or even more than that. And then on the other end, you've got people who are ultra rapid metabolizers, where they might have an amplification of the genes of interest or just have a very high functioning variant. And in those cases, the drug will fail because they will break it down before it achieves a dosage level where it has a physiological response. So for them, it doesn't work at all. And again, a couple of canonical examples. There is a case where, and this is one of the first examples that brought pharmacogenetics to sort of the forefront. There was a case where a woman got a C-section and she was taking, I believe, codeine for pain management, and she was breastfeeding. She was an ultra slow metabolizer, and so was her baby. And the intermediate breakdown product of codeine is morphine. And so morphine was accumulating in her system, and because she broke it down very slowly, and it got into her mother's milk, and the baby suffered from an opiate overdose. And so, you know, what was discovered was, again, both of them were very slow drug metabolizers. And so that's an example of that. And again, the canonical example of an ultra rapid metabolizer is, you know, someone who takes antidepressant, or they take a birth control and they break it down much more quickly than is required for it to be physiologically useful. And so you have this failure of the medication. So here I just pulled the information from the FDA. Drugs, how many drugs are known in pharmacogenomics versus therapeutic area. And so you can see that there are a vast number of drugs in oncology down at the bottom, followed by psychology, neurology, infectious diseases, anesthesiology, cardiology, and so forth. So this is sort of medications versus therapeutic area. But this slide doesn't really factor in the frequency by which these drugs are prescribed. As sort of a general rule of thumb, about 80% of the most common medications that are prescribed in this country, the US, are impacted by the patient's genetics, by their underlying genotypes. So, you know, for example, even though there's relatively few drugs listed in cardiology, there are statins and warfarin and some of the most commonly diagnosed drugs, most commonly diagnosed medications used in that field. So it's a little, so while there are, you know, again, many drugs used in cancer, the frequency, this isn't mapped by frequency. Also, there's just one contraceptive, Yaz, or drospirenone, on that list. And so I think, again, there's a huge opportunity to make improvements in this research space. So this is just FDA Section 1, Pharmacogenetic associations for which the data supports therapeutic management recommendations. And here you can see there's 13 genes that have made this list. There are numerous drugs that are listed for each gene. And the takeaway here is that the potential side effects are specific to the mechanism of action of both the drug and the genetic variant. I'll talk a little bit more about BCHE. It's an anesthetic, and sort of how this works out in a little bit. And there are many, many, many more genes that impact drug response that have been identified at a research level. But optimizing dosage based on genotype is actually a pretty high clinical research evidence hurdle to cross. So there's actually relatively few genes that are listed. Many, many drugs, though. I just wanted to briefly show here some of the models of PGX Action. So up here, you've got this drug, you've got CYP2D6, which I'll talk about in a little bit. It's used, again, it's used in a lot of it is a gene that impacts many, many different drugs. So here we're looking at coding dosage versus gene function, sort of how that interacts. So here's an example where someone might have increased function. There might be there might be a rapid metabolizer, and so for them to get any effect from coding dosage, they'd want a higher morphine concentration. If they had normal CYP2D6 function, then normal function would be fine, and a normal dosage would be just fine. If they had reduced function, then they would need a lower morphine concentration again to get the same pain relief effect. And if they had low or no function, then morphine would be a terrible idea for them, right? And again, you can find this out on the front end before they take the medication by doing the genetic testing rather than waiting on the back end to see what happens, right? Here is a drug called TPMT, which is sorry, the gene is TPMT. The drug is 6-mercaptopurine. It is used for transplant rejection, but it has a risk of hematological toxicity. So if somebody has reduced, if the gene TPMT has decreased function, their increased risk of hematological toxicity increases until it's very high. And so again, you can do front-end screening and identify people who are good candidates for this drug and who are bad candidates for this drug, and provide that information on the front end. The picture over here is just about a gene called, it is about a anti-leukemia drug called methotrexate and how it works in what's called the MTHFR pathway. MTHFR is another gene that is involved in pharmacogenetic regulation because that pathway, this thing called the MTHFR cycle down here, is what gets inhibited by methotrexate. And if somebody has reduced MTHFR function, that pathway moves really slowly, and so methotrexate will accumulate. And this slide is from a study on adverse effects of methotrexate on pediatric leukemia patients. So you've got kids and you want to reduce the amount of nausea and headaches and muscle aches, and vomiting from them, and knowing these kids' genotype, knowing what the status is of their MTHFR's ability to function, gives information about appropriate dosing of methotrexate so that they can have a better time of that particular anti-cancer treatment. So here is section 2 FDA, also use section 3 in a second. Pharmaco-associations force the data indicates a potential impact on safety or response. So here they just know whether there's a high risk of adverse reaction or not. There are 10 genes. They're mostly the same ones from panel A, but with additional drug effects that have been identified. So here, I'll just walk you through it. There's this drug called efavirenz, and it's an HIV antiviral. And if somebody has the poor metabolizing genotype for the gene CYP2B6, then they have a much higher systemic concentration, right? So it floats around in their system longer, and so they have higher risk of an adverse reaction, right? An adverse drug response. That's how that works. So this slide just shows FDA Section 3, the potential impact on pharmacokinetic properties only. So this just speaks to if somebody is a ultra-fast metabolizer or a slow metabolizer, maybe they're going to want higher or lower dosage recommendations. And we, as a laboratory, we just sort of provide that information, right, where it's known. So these 6 genes, you'll also see them in the other lists. There's only a handful of genes total that are kind of listed for FDA, but again, they cover about 80% of the drugs that are prescribed. So just a quick walkthrough. Coliceprolol is a muscle relaxant, and if someone has variants in CYP2C19, here they might be a poor metabolizer, and so this will result in higher systemic concentrations, and so for these folks, they might benefit from a lower dosage. Our PGX panel covers kind of all of the genes that are listed in FDA sections 1, 2, and 3. So we tend to go with the highest level of evidence and the highest level of approval, while hoping that more of them get added to that list. I also wanted to talk a little bit about, again, in the future, this is just absolutely going to explode in utility and complexity over the next few years as more genetic information becomes available. I wanted to shift gears a little bit and talk about kind of how some real world applications to this and some of the other panels we offer. So I'm going to discuss briefly genes associated with anesthesia response. This is an LDT we offer. Adverse anesthesia response impacts about 1 to 2% of patients, so it's less common than the total adverse drug responses, but it can be quite severe. Of which kind of the best characterized is called malignant hyperthermia. So this is a situation in which someone gets exposed to an anesthetic, they have a severe reaction, and they essentially start to cook internally. They rapidly increase to a dangerously high body temperature. Their muscle cells can start breaking down. They have rigid muscles or spasms, accelerating heart rate, and other symptoms. And without prompt treatment, complications can be fatal. So, essentially they start having a massive heat stroke. And again, it impacts, depending on population and other things, about 2% of all patients who undergo anesthesia. It's common. BCHE, which I think was the top of the table 1 in the FDA, because it's very, very early identified. So succinylcholine is a drug that's used for facilitating tracheal intubation in emergencies or for patients with a high risk of vomiting. Basically, it temporarily paralyzes the throat and the respiratory muscles so that they can get the tube in. Some people are, and it does it fairly quickly. They go under it paralyzes them very briefly. They get the tube in and they breathe normally. But some people are slow metabolizers, and so they are at risk of asphyxiation because of prolonged incapacitation of the respiratory muscles when they get a normal average dose of succinylcholine. And again, it's incredibly useful to know in advance what that response is going to look like and what the optimized dosage is going to be for them. So it was one of the first pharmacogenetic disorders identified. And the other thing to say is that there are over 60 SNPs in BCHE that have been reported. So some of which have functional impact, some of which that don't. And so a sort of a small panel isn't going to capture that. For these guys, you're better off just sequencing the entire gene and catching any risk markers that are out there. There are other malignant hypothermia, anesthesia responsive genes, CAC and A1S, which is in our panel. It's, again, a susceptibility to malignant hyperthermia gene. They're both calcium channel encoding genes, and so I guess the mechanism of action is that the medication kind of uncouples these channels and starts this kind of chain reaction that starts the muscles locking and spasming and then cooking. It's strongly actionable for a number of different drugs. And again, the variants are spread throughout the entirety of the gene. So similar to breast cancer 1, the one gene, you're just better off sequencing the whole thing to identify any risk variants. So, one of the complexities is that when you do whole gene sequencing, so ROR1 is associated with malignant hypothermia, but it's also associated with other conditions. Here, RYR1 in the green box is also associated. It's associated with malignant hyperthermia, RYR1 related myopathy, so sort of muscle diseases, sarcopenia, fatigue, myasthenia gravis. So you can end up in a situation where the doctor or patient gets more information they expected as a function of testing in terms of familial risk. And in some cases, genetic counselors are just a critical component of these sorts of conversations. So looking at where this testing is going, here I think you can take a page from cancer genetics, where there a number of genes have been identified that are associated with increased risk of cancer, but these genes are modified by a wide variety of other by a number of different other actors. There are upstream signaling mechanisms, receptor variants, there are transcriptional regulators, there are translational regulators, the signals that these provide, like, they send the message to downstream genes or downstream proteins as part of a complex signaling pathway, and modifications in any of these systems can result in big differences in the function of these cancer genes, and that's probably going to be the case as well with genes involved in medications. So again, there's lots of different ways to turn up or down the function of any given gene. And here we're just looking, like what we're currently doing is we're just looking at genetic variants that impact function within that single gene. Whereas we know that there are some regulatory systems that can turn up or down, like a volume control or a light dimmer switch, the concentration of these genes or their ability to get turned into proteins and so forth. So here I just posted a couple different slides looking at some of these regulatory factors. So CYP2D6, again, very important for drug metabolism, and it has known transcriptional activators, right? Retinoids induce the expression of these regulatory elements. Pregnancy can impact them, and this can result in reduced expression of some of these transcriptional activators. And so these will modulate the amount of CYP2D6. So you can imagine a situation where someone has a normal, they make a normal CYP2D6 gene, but they don't make very much of it, right? Because one of these genes that are upstream of the activation of this gene, one of the activators doesn't function that well, or one of the repressors works super well. So once you get sort of in the regulatory weeds of these genes, there's a lot of different ways that their activity can be modified. We know this from cancer, and so when we look at pharmacogenetics, we are going to identify more of these over time. There are splice variants within these genes that have been identified. What are the impact of these on pharmacogenetics? Don't know yet, right? CYP2D6 is strongly associated with both drug metabolism and schizophrenia. So are variants in the expression going to have effect? Are there different genetic factors at play here? Are the different splice forms of these genes going to be relevant? We will find out in the years to come. Maybe neurotransmitters are being rapidly metabolized in the system. We just don't know. Finally, just again, another example. It's a pretty noisy slide, but my only point here is that there's a world of interacting regulatory factors for pharmacogenetic associated genes, and some of them are quite novel and quite subtle. Here, there are these things called, micro-interfering RNAs that bind to the RNA that gets produced by these genes before they get made into protein and downregulates them or inhibits their ability to get turned into proteins, so that the amount of these genes can be upregulated or downregulated by these other factors. And again, more, since I made this slide, more microRNA regulators have been identified. So these are going to play a role in how much of this protein is around. So you can imagine a situation like where you are a high, you have highly functioning CYP2D6 proteins. You're a high metabolizer, but you have a microRNA inhibitor that is also high functioning, and so it prevents you from making that much of this. And so you're in effect a normal metabolizer, right? Those sorts of questions and the ability to ask those sort of questions and make those sorts of clinically relevant calls are what's going to be coming up in the years to come. 100%. So very briefly, I just wanted to say there's been many studies to look at the cost effectiveness of PGX testing. I sort of squinted at them, and it looks like for the average patient, getting this testing done saves a couple grand a year for each year going forward that they're testing. The estimates range about $1,000 to $3,000 annually if you sort of squint and look at the big picture. However, in specific cases, there are enormous benefits to PGX testing. So it turns out getting someone with suicidal ideation on the right antidepressant quickly has enormous benefits in terms of sort of the economic measurement, which is called quality adjusted life year. So in the Canadian health system, which is sort of the second bullet, PGX-assisted treatment for depression cost effectiveness ranged from $12,000 to $16,000 per quality-adjusted life year, right? Preventing someone from killing themselves by getting them on the right antidepressant quickly turns out is enormously beneficial. Some cardio drugs have a small but significant savings, and some of them are huge. So I guess the fourth bullet down here, prasugrel for antiplatelet therapy on a large number of patients studied, looking at uncontrolled bleeding, unguided treatment, resulted in a cost of about 40,000 pounds in this study, and this number was brought down to about 8,000 pounds when they did PGX first, and they removed the known high-risk people from that treatment, or they went to an alternative treatment. The simple economic analysis can be confounded because in some cases you're taking somebody off a cheap drug because they're going to have adverse effects. So they're going on a more expensive drug, but they're having less adverse effects. So the health economics doesn't necessarily capture the quality of life improvements associated with making better PGX-informed medication decisions. ARUP's laboratory, very briefly, they tested their own staff. So they had 228 voluntary participants. ARUP Labs is a fantastic diagnostic laboratory in Salt Lake City, and they found that of their staff, about 54% of them were taking medications that were going to have adverse effects on them. So there were 54% of the recommendations were to use a different medication than the one they were on. Again, in average population, they were fine, but no individual is average. And when you factor in someone's genetic individuality, or their biochemical individuality, there are lots of opportunities for improvement. Of those recommendations that were made, 41% were related to psychiatric medications, about a third were related for chronic pain, getting those dosage optimized, and 13% for cardiovascular drugs. They were trying to do a long-term study on the economic benefit, but they did the study during the year of COVID, so it's kind of things kind of went off the rails. As I remember from their presentation, they were looking at something like 10% annual improvements in terms of in terms of healthcare costs. Doing it might have been absenteeism. So again, the summary here is that a preponderance of evidence suggests that PGX treatment is cost-effective and often cost-saving when considering treatment with medications that have known genetic interactions. As is apparent in psychiatry, PGX testing is especially invaluable where multiple medications with well-documented associations are considered. There are challenges in terms of implementing PGX. There's a sort of regulatory hurdle where there's, sort of research galloping ahead of approval, and developing testing has its own challenges, sort of LDTs versus kit-based testing. There's challenges in terms of study design that I think sort of got alluded to in the previous slide. You have to do PGX versus each medication, and you have to define your outcomes clearly. There's differences in study design and in patient populations. Some new drugs, you may not know which metabolic pathways are involved, and there's obviously all sorts of emergences. There are many new clinically significant genetic variants that again have sort of galloped ahead of the sort of regulatory piece here. There's challenges in clinical implementation, for these custom testing panels. You have to, there's compliance in terms of ordering and interpretation and billing. There's institutional inertia. You have to make sure that there's sufficient evidence and utility, and again, FDA approval. And obviously, there's patient privacy concerns about how these results are used properly. Sort of for us, we focus on a couple of things. Obviously, our main focus is on clinical utility and compliance. Does the test inform the diagnosis or treatment? We generate an enormous amount of information through this testing. If you sequence a whole person, you get about 4 terabytes of data. But the clinician doesn't only needs the information that is actionable in terms of diagnosis and prognosis and treatment. So we, while as geneticists, we get to geek out on this information. It's super interesting stuff. For the clinician and for the patient, they only need to know what is important to them. And so spending a lot of time on curation, making sure that the actionable information is what gets to them so that people in a hurry can do what they need to do and get on with their lives. Right? There are many different working groups, and different consortiums that compile this evidence, and it's changing all the time. And so it's important to stay current on that. And that's another thing that we focus on here. So this brings us to the opportunities and challenges of personalized medicine, right? On a prevention level, you can detect patients at risk at an early stage and improve their preventative measures, right? If you know you are high risk for taking a medication, for example, you can take something different if the alternatives exist. And that's again the sort of information that a good report can provide. You can get accurate disease diagnosis, enabling individualized treatment strategies specific to the underlying pathways involved. And then finally, you can improve outcomes through targeted treatment and reduce side effects. So that's the payout of moving towards a more personalized medicine paradigm. Finally, again, I admit I'm a little biased as a geneticist, but since precision medicine saves money, I think it's only a matter of time and data accumulation before this becomes standard of care. It's just too common for someone to have a variant that is going to impact their treatment outcomes. Right? If you're trying to reduce adverse drug responses, if you're trying to take effective doses, not get these little overdoses or not get underdosed of a medication, it's just these genetic variants that impact that decision are just too common. So that's where I think this is going. In the slide from NERDS 2018, they sort of say current reactive medical care model. I kind of think that's a little heavy-handed. I think what we're currently doing is empirical medicine. You make the diagnosis, you try the therapeutic, people come back, you find out if that worked, you switch and switch and switch as needed until you get it right. Here in the personalized medicine paradigm, you get screened on the front end, and then you know which drugs to avoid or what optimal doses are going to look like, and then you can monitor from there. So that's where I think that's all going. I think there's going to be explosion in risk and severity markers as human genome testing becomes more ubiquitous and more cost effective. The prices have gone down from billions to thousands to about 1,000 to lower than that. So I think we are going to have an explosion of genetic information, and then the focus for us is going to be on clinical utility, getting actionable information to patients and clients and clinicians so that they can make those better decisions and improve their healthcare outcomes. I put my email up there if you have any questions, and I want to thank you all for listening in, and I hope you've enjoyed this webinar.
Thank you, Jonathan, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, let's go ahead and get started. Our first question is, where do you see the future of pharmacogenomics going, and how does NGS fit into it?
Oh, that's such a great question. Thank you for asking it. I think cancer is the best model for this. So all the known cancer genes have regulatory elements, promoter variants, interfering RNA upstream and downstream functional modulating genes that are going to likely impact that we know impact outcome. And my guess is that that's going to hold true for pharmacogenomics as well. Again, I think there's going to be, eventually we'll have a comprehensive sort of whole genome NGS approach towards this, and we'll have identified many more variants that maybe are rarer, right? Maybe they impact fewer people, but for those individuals, it's incredibly important. And those will get sort of added to the list over time. Right. And again, the frequency of PGX variants or such, I think, is going to be standard of care. And, I wanted to slam the FDA for being slow to get these tier 1 evidence genes approved. But, the sort of PharmGKB, the sort of big database, lists, there's about 873 drugs that are known to exist that have annotations based on genetic variants. And the FDA lists about half of them. So I don't actually think they're doing that badly on that front. But I mean, again, there's twice as many drugs out there that sort of haven't gone through this evidentiary process. So those are all coming. Those will all come with time. Research tends to gallop faster than safety, and then sort of FDA, and that sort of makes sense. The FDA is associated with safety, and research is about finding stuff out. So I think in time, one will catch up with the other, and we'll have a lot more information to give to people.
Okay, great. Thank you. Your next question, what are the most important considerations when choosing an NGS platform for pharmacogenomics testing?
That's a great question too. I mean, there are, there's a couple things going on here. There's clinical considerations like assay flexibility and accuracy and reproducibility. And then there's also the business considerations, also assay flexibility, because again, as research identifies new markers, you want to be able to add them very quickly to your testing panel, right? As new genes get identified or new drugs get pulled in, you need these sort of you need the flexibility to do that. But again, you also want your setup and your per sample run costs to be relatively low, and you're going to need a high level of support because these are pretty complex to set up. Turnaround time is important. Throughput is important. Financial flexibility in terms of leasing versus buying, and all those things need to be balanced when platforms are considered. But those are the big ones for me.
All right. Your next question. Where do you see the value of NGS as a modality in the space above other technologies like PCR?
Oh, okay. So this is kind of a no-brainer. Some of these genes have, specific hotspot SNPs, and you can do PCR to individually identify them. But in many of the genes I showed, BCAG, RYR1, the variants, the genetic variants that people have, are spread across the entire gene, right? So in those cases, NGS is going to be way better in terms of providing comprehensive coverage of risk identification. And because these risk alleles are dispersed through the entire gene at a low frequency, you definitely need an NGS platform to identify them, right? The alternative is you're amplifying hundreds and hundreds of SNPs, and that's going to change over all the time, and very rarely will one of them show up, as opposed to sequencing the entire gene and identifying the variants that are present.
All right, thank you, Jonathan. We have time for a couple more questions. Your next question, for labs looking to enter the pharmacogenomics space, what advice would you give them to help them succeed?
Oh, what a great question. Okay, so when my current lab was getting started, when Tesis was just getting started, we definitely benefited from working with outside experts to validate our systems, our assays, and our reporting. So next generation sequencing is a very B2B collaborative endeavor. Like, you are definitely going to want to have dedicated experts who really know how to do it. And also, do not skimp on IT support. A single whole genome can require 4 terabytes of information storage, and PGX runs will fill up your platform storage quicker than you can imagine. So you need to plan it accordingly in terms of on-site or cloud flexible storage, right? Again, the actual running gets very routine, but the setup can be quite challenging, and you're just far better off bringing in experts to help that out.
All right, thank you. And your last question, around pharmacogenomics, what lessons have you learned over the years that you wish you knew sooner?
Okay, so I'll do two. So I used to joke that that they called it next generation sequencing to make folks of us who used to do silver staining silver staining gels and counting nucleotides feel old, but it's actually become hugely simplified, right? Again, the startup is really complicated, but once things are running, the process truly looks plug and play compared to back in the day. I compare it to using a toaster versus building a toaster from scratch. So, so don't be daunted by that. Just bring in experts. That's just the way to go. And also, I should say these systems, they generate mountains of genetic information, but clinicians only need to know a small fraction of it, which is the parts that are clinically actionable. So wherever possible, bring in people who know how to curate that data. So you're not drowning clients in information that just isn't relevant. Um, telling someone that they have a genetic variant of uncertain significance does them absolutely no favors, right? Whereas telling them, here are the pathological variants and here's what is typically done with them, providing that information, that is something people can use. And it's all about providing value to people.
Thank you, Jonathan. Do you have any final comments for our audience before we close today?
Sure, I say this all the time, but we are in a golden age of biomedical sciences. We are going to, again, as the costs of whole genome sequencing and whole exome sequencing get lower and lower, we are going to generate so much clinically actionable information that helps people make better decisions about their lives and improves the opportunity for them to live longer and healthier lives. So it's a wonderful thing to be part of, and I just want to thank everybody for your time today.
Well, thank you again, Jonathan, for your time today and your wonderful presentation. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.